Hi, welcome back to Father-Offspring Interviews, episode 49, sadly no longer in person, as you can see. Um, so first, Wayne from Georgia says, a friend recently passed away after a hopeless battle with Creutzfeldt-Jakob disease. I was just wondering if you know of any medical treatment advances or studies that have been able to give more insight to a possible treatment in the future. It seems very hard to study since most patients, from what little I've been able to find, tend to die within 12 months of being diagnosed. Also curious about how it transfers to offspring and what their chances of falling victim to the disease are.
Well, to quickly answer those, but to start off with, apologies. I have a cold, so I'm going to be rumbling here somewhat. To immediately answer some questions, no, there's no treatment, there's no cure. It's 100% fatal, typically within a matter of months, tragically with your friend, and unless very, very rare, it is not transmissible to offspring. What it is one of the nightmare diseases. One of those you really do not want to get, amid all the other diseases you really don't want to get. It is catastrophic. And just to sort of get a tone you're not supposed to, unless you're a scientist, it's crazy interesting. It is such an interesting disease, and it's a class of wildly unexpected diseases that just puzzled people forever. Class of diseases now known to neuropathologists as spongiform encephalopathies. There's Creutzfeldt-Jakob, there's fatal familial insomnia, there's scrapie, there's mad cow disease, there's kuru, there's a whole family of them. Where is this name from? Spongiform encephalopathies. That's just descriptive when somebody dies of Creutzfeldt-Jakob, of any of these, and you take their brain out and it's sliced up and you're ready to examine it. And if you're in the business of examining sliced up brains, what you're accustomed to is looking under a microscope. Ooh, look at that. Here, this bunch of neurons have died, but those neurons look just fine. Or all these neurons have died, but the glial cells around them look just fine. Selective cell loss. In Creutzfeldt-Jakob, along with these other diseases, there is no selective cell loss. It is like a bomb goes off inside your brain. Neurons everywhere are dead. Glial cells, the other main type of cell, they're dead. Blood vessels are dead. Just an explosion of non-specificity. And here's where the sponginess comes in. This is so extreme that even without a microscope, you can see holes all over the brain, like a sponge, like a sponge. And then another term that got introduced into neuropathology around then, it could be so extreme that you see cavitation of the brain, cavities of empty space, and it collapses in on. Okay, so all of these are spongiform encephalopathy diseases, which is a fancy way of saying it is the most terrifying that a brain could look post-mortem after any disease anyone had ever seen. So, where is it coming from? And for this, we've got to go back 70 years now, historically, to where all of this started. And it is an amazing story that I'm going to just go over somewhat lightly because the details are numerous, but add to the fascination of it. But it had to do with the discovery of another spongiform encephalopathy, Kuru. Kuru, some people may know that it's as exotic as neurological diseases get. Kuru is only found in one tribe, the Foray people, in the highlands of New Guinea, and it's an infectious disease. This was first really studied by an American neurologist named Carlton Gajicek. Gajicek, one of the geniuses of the 20th century, one of the eccentrics of the 20th century, a terribly, tragically damaged man who wound up being tragically damaging on his own. But Gajicek in the New Guinea highlands spent years trying to figure out what this disease was about. And just to give away the punchline is it was an infectious disease. There was an infectious agent which got into people and then caused kuru, and your brain looks like a sponge. And as soon as you start getting symptoms, you're gone within months. But here was the thing about it, which is eventually the punchline, the like how this hasn't been made into a dozen movies by now. The punchline, what was the route of infectivity? The 4A people were cannibals, not exo-cannibals, which is, the horror movie version of like you capture your enemy and rip out his heart and consume it to get his strength, which turns out to be overwhelmingly a myth in the anthropological literature. What the foray were an example of people who were endo-cannibals. Your mother dies. And as your sign of respect, you cook her up, and the entire village comes and eats her remains. This is what is done. This is what any child should do. This is what any parent expects of their children for after they're gone. Endocannibalism. And it turns out the infectiveness of a Kuru brain, someone who had died of it, was transferable to the next person. The person not who ate the brain, but the person who prepared the raw brain material. The daughter, typically, of the person who had died, the daughter who would be a young adult. And this was what it was about. And how that transferred is a whole story there, but it was transmissible. It was coming from handling raw infected brain tissue from your parent that you've taken out and you were about to cook up. And at the time the Gajasek came into the New Guinea highlands, Kuru and the clan retributive violence for the witchcraft that obviously was the cause of Kuru collectively were one of the leading causes of death there. So there were a lot of ceremonies of people who had died of Kuru. And thus the young adult woman handling the brain material would get infected with it. How she did, details there, but the detail that floored everyone is young women didn't get Kuru. Middle-aged women didn't. Elderly women did. Elderly women, decades after they had cooked up their parent at this cannibalistic ceremony and handled the raw brain tissue. So, Gajasek slowly getting evidence that this was an infectious disease, some kind of virus, and this made no sense whatsoever. Virus, you get a virus and, 2, 3 day incubation period for a coronavirus before symptoms start. And what Gajasek was piecing together was the incubation period for this infectious virus thing was years, was decades. Ultimately, the record case is more than a 50-year incubation period between when someone was exposed to Kuru brain tissue and when they themselves finally developed Kuru. This was simply unprecedented. Nobody believed him. And actually, what it took was eventually him coming with purified plaques, the part of the brain tissue in people with Kuru that contain the infectivity, and injecting this into lab animals and showing that after a long delay, they developed something like Kuru. It's infectious. It's infectious. Okay. So this is totally wild. Gajasek gets a Nobel Prize for this, for the discovery of slow viruses, but still nobody understands how this works. And the story proceeds to get even stranger at this point. And this revolves around a scientist named Stanley Prusiner, who at UC San Francisco, Prusiner, who starting, I don't know, in the early 80s, was trying to bring molecular biology to it. What's this infectious agent? What is this thing, this thingy, this virus, whatever? And because Kuru brains were very hard to come by, what he focused on was the sheep equivalent, a sheep and goat spongiform slow viral disease called scrapie, which he could get large amounts of. And he started purifying the little plaques of aggregated infectious particle that you would find in the brain afterward. And he slowly began to piece it apart. and slowly began to show that it was infective, like a virus is, and slowly hit a wall of not being able to make any sense of this. Okay, viruses. What a virus does is invade a cell and take over the cell's DNA and the cell division replicative machinery for its own purposes. So your standard issue virus is made of DNA and comes in there and takes over. And around the time Prusiner was showing up, people were just dealing with this trauma of dealing with a new type of virus, something called an RNA virus. Okay, central dogma of life. You got DNA coding for genes, which code for RNA, which code for proteins. So evidence that there's RNA viruses, and this was totally impossible, and people speculated, what, are you saying anything's ridiculous as there's something that could turn the RNA virus back into a DNA virus and then it's infected? That was the only possible explanation. And people found an enzyme that did exactly this, a reverse transcriptase, and that earned some Nobel Prizes. And so we've now got DNA viruses and RNA viruses. And Prusiner, who had now been purifying this infective particle and transferring it to lab animals, all of that, and showing this was it, this was the infectivity, had this impossible finding, which was there was no DNA in it. There was no RNA in it. There was just protein. This was a protein virus. And, I was in grad school at the time and was at a meeting where he presented some of the stuff. And like everybody just threw their hands up in the air. Like every old time virologist was saying, like, damn, viruses are made of DNA, and we just overcame the trauma of dealing with the fact that viruses sometimes are made of RNA. Now you're going to tell us that like viruses are made out of protein? No way, impossible whatsoever. And Prusiner was mocked with skepticism for years with the assumption that when he was purifying this infectious particle, somehow in the process, he was washing out the DNA or washing out the RNA and was just left with the protein that was kind of like the styrofoam packing over the virus. And that's what everyone threw at him. And he just showed again and again, and come to my lab and work right next to me at the bench, and you will see, you will purify it the way, and just fighting his way through. And what it finally took was showing you make a synthetic version of this protein in a test tube and you inject it into a guinea pig, and it develops a guinea pig's version of scrapie. It's the protein. It was really the protein. And Prusiner came up with a great term to describe this. He said these were prion viruses, prions, proteinaceous infectious thingies, and prions were protein viruses. This was undeniable at this point. And everybody who was finally willing to admit it was protein, still now, many years later, there's a few holdouts. Most of the field said, yeah, this is a protein virus prion thing. They're saying, what? Are you now going to tell us that the protein infectious agent gets turned back to RNA and then gets turned back to DNA and is infectious from there? And this was just outlandish, and this was impossible, and this was the last straw. And then Prusiner came in with even more of a last straw as he finally figured out what the mechanism was for the infectivity. He isolated the protein that was infectious, the scrapie protein, because these were scrapie brains. The scrapie protein had like a distinctive shape to it. And it turned out it looked kind of similar to a protein that occurred in cells, a normal cellular scrapie protein instead of the infective one. They were sort of similar structurally, except the normal one was straight. The normal one was like, where's a different conformation for the prion one. And what he showed was that if one of these normal cellular prion proteins bumped into an infective scrapie one, the infective scrapie one forced it into the same conformation. And now it was two of them bound together. And they encounter a third one and a fourth one and start building this aggregate. And that was the mechanism for what was happening. This was outrageous. This was totally wild in terms of a mechanism of like, okay, so some sort of mutant bended prion scrapey protein doing it. Anybody who ever read a Kurt Vonnegut novel, oh, what is it called? Cat's Cradle, which I read in high school and was greatly influenced by along with the rest of his writing. Cat's Cradle. In it, there is a reclusive, brilliant scientist who's off doing his experiments, and he's always being bothered by reporters wanting to know what he's working on. And a reporter comes in, and the scientist is in this sort of malicious mood. So he makes up this total story. He says, actually, I'm working on this great secret project. I'm making a new type of water. You know, water has two hydrogens and one oxygen, H2O, and it has a certain conformation. I'm making a type of water which has a different conformation. And I call it Ice 9 because it's the ninth one I came up with. And if a piece of Ice 9, one molecule of it, bumps into a regular piece of water, it forces that water into the same conformation, which forces the next, forces the next. And the reporter said, "Oh my God, that's amazing. But what if it fell into the ocean or something?" And he said, "The entire ocean would freeze and would be the end of life. Get out of my lab." So, okay, so the guy leaves. And of course, what the novel is about is the chemist actually has made a piece of Ice 9. And on his deathbed, he divides it with his three kids, and each one of them gets one in a thermos. And the book is about what they do with it. And at the end, a piece is indeed dropped into the ocean by accident, and all of water on Earth freezes, and it's the end of life. Infectious, scrapey proteins are like Ice 9. You get one of them in there, and eventually, inside one cell, statistically, 5 years later, 20 years later, it's going to bump into one of the normal ones and turn it into that. And because two of these now, what's called a dimer, two of these now are bigger than one alone, it's a little bit more likely that they're going to bump into a third one 4 years later, and then bump into a fourth one a year later, and then bump into a dozen of them 6 months, and then bump into 100,000 in the last month in this exponential explosion. And what you saw was one single particle in the brain would slowly go about, eventually turning the brain into a prion crystal, crystallizing outward like Ice 9. And that's why it took decades and decades until it bumped into another and another and another. And one of these managed to get into the next neuron. And and the next neuron and the next neuron into the brain from there. So this was this completely unprecedented process. Then the final sort of bombshell from Prusner. So what's the obvious thing? Well, the normal prion protein, the normal scrapey cellular protein, should have a certain sequence of amino acids, and that makes it up, an alphabet that forms it. And the abnormal version obviously was going to have one of the letters wrong, was going to have a mutation right here, which caused it to have this different. It was a mutation because that's what mutations are. And what Prusiner announced was no, there's no mutation. The letter sequence that makes up the normal protein and the abnormal one were identical. This was not a mutation in the gene. It was the exact same sequence. That's impossible. Everyone said who had spent the last 20 years saying it's impossible to anything that Prusiner or Gadjicek had been saying. Because once again, the central dogma of life, DNA coding for a single strand of RNA, which specifies a single strand of amino acids, that make up a single protein, which has a single conformation, which has something to do with its function. One gene, one RNA, one protein sequence, one conformation, one function. And here's Prusner saying, no, the exact same amino acid sequence. Some of the time it could be perfectly normal, and some of the time it could be in this weird pathogenic thing. And this ushered in this whole challenge to this central dogma of life. The exact same protein, the same amino acid sequence could have multiple conformations. So the question becomes, what's the current status of these diseases? Kuru is about to disappear. Probably the last case was 20 years ago because the cannibalistic ceremonies were given up in the highlands there. So that's about to disappear. What about this scrapie prion disease that you see in goats and sheep? It's something that pops up in ranches now and then and is a misery, and you have to kill the whole herd and all of that because it's going to jump from animal to animal somehow or other. Saliva, it's not clear, and something like scrapie. And then about 35 years ago, through very unclear circumstances, the scrapie infective particle jumped a species barrier. It jumped from goats and sheep into cows and invented a new disease there called bovine spongiform encephalopathy, mad cow disease. And mad cow disease was the cow's equivalent of goat and sheep scrapie. And as you'll perhaps remember, before it was over with, I don't know, 200,000, a million cows in the UK had to be slaughtered to stop the spread of it, to make sure this did not get in the meat supply. All meat, that's muscle. What's that have to do with the brain? Muscle has nerve endings in it. And that it wouldn't get into the food supply, and complete panic. And there was every reason to think that this was going to get into the food chain and this was going to get into humans, and who knows what it was going to do. And we soon found out what it was going to do. In humans, it produced something that looked like Creutzfeldt-Jakob disease, what came to be called variant Creutzfeldt-Jakob. And the characteristic of it was incubation was way fast. You went from exposure to the disease in a matter of months or whatever, forget 50 years. And who was getting it? As it turned out, a bunch of people who were eating 400 McDonald's hamburgers a day in England, kind of think heavy, heavy beef consumption. And everybody wet their pants in a panic at this point, and we dodged the bullet. There's been, I don't know, 20, 40, 50, 100 cases of variant Creutzfeldt-Jakob. It didn't get into the general food supply. It could have affected millions of people. It could have made COVID look like nothing. Sheer random, utter luck that that didn't happen. Okay, so that's what that's about. So we see the very rare cases of Creutzfeldt-Jakob from these hamburger obsessives in England in the 1990s, where thank God it didn't spread. And there's some horrifying sub-stories about that that emphasize how easily that could have happened. Once again, sheer stupid random luck. So we see some people can get Creutzfeldt-Jakob from hamburgers, very, very rare. Very, very rare. There are some individuals who spontaneously have a mutation in that gene so that it normally takes on this conformation, and off you go, and that's going to kill you at a very young age. But in theory, if you reproduced, you could pass on that mutation to your offspring. But stupendously rare. Where do most of the cases come from? And here we introduce a very interesting word, a word that makes medical people queasy with the implications of this, the word iatrogenic, which is diseases that are caused by medical procedures. And here was the secret with prions. Scrapie prion, Creutzfeldt-Jakob prion, Kuru prion. They are the most densely packed molecules, aggregate hairball cruds of cross-linked everything, it is virtually impossible to break them down. So you're doing brain surgery on someone who's suddenly having a rapid dementia and you're wondering what's up, and you go in there and you conclude afterward when they die 3 months later, oh my God, it was Creutzfeldt-Jakob. That's what was going on. But fortunately, after you did the surgery, you took your surgical instruments and you sterilized them. You sterilized them exactly as you're supposed to do, and sterilizing didn't touch the infectivity of prions. Radiation didn't do it. Bleaches didn't do it. Just this whole sort of protocol now that's needed to break them down. It is next to impossible to break them down so that the surgical instruments you used pass them on. The dental drill that you used passed them on. And people began to identify clusters of cases of Creutzfeldt-Jakob where it was spread in this manner. One particular case in the 1980s, kids who were having impaired growth, there was this whole program at National Institutes of Health where growth hormone was extracted from cadavers, from the pituitary of cadavers and purified, and then kids were injected with growth hormone, and it was very helpful. And somehow in one of those batches, someone who had donated their body had a case of Creutzfeldt-Jakob, which somehow wasn't spotted, and it got into one of the preparations, and this spread Creutzfeldt-Jakob to a whole cluster of kids. Interesting side story, that whole program was shut down the next day. You could not get purified human growth hormone anymore. And out of luck, and a couple of postdoctoral scientists at UCSF at the time looking at it, who studied how to genetically engineer bacteria, said, whoa, what if we could make the bacteria make human growth hormone, which was the first gene engineering. And they got it to work, and they decided to set up a little company on the side, which became Genentech, the biggest biotech company in the world at this point. So that's where that came up. Okay, so iatrogenic transfer. There's a layer wrapping around the brain called the meninges, and you can have transplants of meninges from corpses to someone who needs a donor. That turned out to be a vector for it. Again, root canals. Take it out there and you sterilize your drill, and this person had Creutzfeldt-Jakob at the tip of their nerve root and there, and you transfer it on there. The majority of cases of Creutzfeldt-Jakob in the United States appear to be transmission in this way. There's a few others. There's a smidgen of evidence that it could be transmitted in milk, that it could be transmitted in a blood transfusion. There's tiny bits of evidence. People who are obsessives about raising championship roses, they use bone meal apparently that you grind up as fertilizer, and it turns out occasionally there's like cow bone meal that has an infectious prion particle in it, so that seems to have accounted for a couple of cases. So that's the main thing. The one other way in which Creutzfeldt-Jakob is most commonly getting into people is indeed through the food supply. Despite the mad cow disease near miss, what you find is there's all sorts of cultures, Mediterranean cultures, other ones, where traditionally there is consumption of goat brains, sheep brains, things of that sort. And that explains a whole lot of clusters of Creutzfeldt-Jakob in those places. And these are brains you cooked. You cooked them up, and that's not enough to undo the infectivity. So this remains one of the truly terrifying diseases. Amid that being all set for the next zombie series, nonetheless, it is supremely rare. Nonetheless, when it does happen and there's an autopsy done, everyone is scared out of their boots and has more layers of protective clothing on them to make sure they don't get infected. Prusner, everyone had to eventually admit he was correct, and he got his Nobel Prize in his 80s now and still researching prion infectious diseases. An astounding story. anthropologically, molecular biology, virologically, yucky brain diseaseology, like a totally remarkable piece of medical history.
All right, now Muhammad from Palestine says, I've read a criticism of your view and your latest book on free will that you're only analyzing it from the scientific standpoint and not engaging with the philosophical literature on the topic. Do you think science alone can or already has fully tackled the problem of free will?
Okay, I'm going to be a totally dogmatic pain in the tush here, in that I think not only can science explain our lack of free will, science is the only thing that could explain it. And philosophy is not relevant to the free will issue. Now, I'm particularly prickly about this right now because I was reading a paper article the other day. It was actually a review of someone who should professionally know better than the conclusion they reached, but someone who I dare say does not know a whole lot of life sciences. And I want to get the quote correct here, but towards the end of the piece said, and I quote, "Science can't prove there's no free will because the question of free will is not a scientific question, but a philosophical one." And greatly irritated, here's the thought experiment I would offer to that individual who gets up one morning and they haven't gotten quite enough sleep because they got a big deadline and they got to work on a project first thing in the morning and they got to finish editing and writing it, and they're like not getting anything done. They just can't concentrate. They get a phrase and then it slips away from them. They keep like messing up spelling. They're so frustrated because come on, focus, focus. This is important. There's a deadline. You got to get it done. And afterward, they finally like get out some crummy, acceptable version of it. And they're frustrated saying, why couldn't I get myself to like do a better job on it? And then they suddenly discover in the kitchen something got moved and they had decaf coffee that morning by accident instead of regular caffeinated coffee. And what that person would just have proved is free will and its lack thereof is not a philosophical issue, it's a scientific one. Every time you show somebody with a brain tumor whose behaviors change dramatically, every time you show somebody who has screwy neurotransmitter levels and they get labeled with a psychiatric disorder, every time you get somebody who has a weird gene variant of something in their brain and they spend their life having trouble regulating their emotions, anytime you get a 5-year-old who, because they happen to be born into poverty, already has a part of the brain blunted by stress. And that's a scientific phenomenon, not a philosophical one. Already at 5 years of age is having trouble regulating their emotions and doing long-term planning. Take all of those pieces together, and science is all there is for proving that there is no free will. No single discipline can do it because it can't control for everything. You study the brain, you have no idea what's happening in the cultural history of the person. You study paleontology, you have no idea what the fossil had for breakfast. But you put all of the disciplines together, and not only do they collectively plug all the holes that the other disciplines have, they all merge to form a single mechanism of explanation, a single matrix of explaining how we are nothing more or less than the biology that we had no control over and its interactions with environment over which we had no control. If you talk about the effects of genes on behavior, by definition, you're talking about millions of years of evolution to those genes. And by definition, you're talking about fetal life where epigenetic programming of those genes occur by environment. And by definition, you're talking about how much of the protein you made from that gene in this part of your brain this morning. And all the pieces come together, and there is not a crack anywhere in that edifice of explanation in which you could shoehorn free will without resorting to magic. So as you can see, I'm very irritated by that quote. Whether or not free will exists is an issue of science, and my belief is it absolutely collectively shows there is none. What we should do about the fact that there is no free will. Now it's time to let in the philosophers from the backyard after you locked them out there. And now's the time where a molecular, epigenetic, neuroendoimmunobiologist probably doesn't have much to say. Now's the time that the philosophers and the poets and the clergy and the writers come in and have something to say. But whether or not we are organisms that would be breaking the rules of how every other biological machine on Earth works and have the ability to will ourselves to break every rule we know about how the physical universe and the biological universe works. How that is not the case is a purely scientific question. So, nice and calm about that one.
And that's episode 49. Keep submitting questions at the form found in the Instagram story highlight and bio or the YouTube video description. I'm Offspring Sapolsky, and thanks for your continued support of Science and the Beard.